السلام عليكم ورحمة الله وبركاته. بسم الله الحمد لله والصلاة والسلام على رسول الله. I'm in such an auspicious moment. I'm too happy for getting an opportunity to present this paper. My paper on the topic Genetic Engineering, CRISPR, Abutment and Development for Medical Science at Current Discourses. I think this current era, it's a specific time to discuss the genetical engineering and its developments. The way is rapidly developing on genetic engineering to its effects. So more discussions may bring more things and more developmental theories in our minds and researches. I will be discussing here on genetical engineering, history, discovery, application in medical field, clinical benefits, and I'm more focusing on the designer babies, where the world will change. We can bring a new era of world where the diseases are eradicated. As first of all, CRISPR-Cas9. The full form is clustered regularly interspaced short palindromic repeats. It is a technology used in germline editing and genetical editings, where it utilizes the Cas9 protein for these purposes. And when we look after the history, before history, just a reminding, where just short note, what is CRISPR? It's just functions a GPS mobility inside the body, where it finds the location of the DNA and it acts as a scissors for cutting the DNA, and it also acts as a pen for rewriting and re-editing the DNA, where we can change the world. We can change the methods and physiological and anatomical purposes in the human bodies and even in the animals. So it is mainly focuses on the RNA or ribonucleic acids, as I think you all know it. And these RNA are the products that are formed from the DNA sequences. So the Cas9 is playing a major role in CRISPR, where it connects with the RNA for editing, and it gives a space where the editing is being possible. And when we look at the history, we all know that in our country there are some breeds of plants, there are breeds of animals. So we the people on more years ago, we were breeding the plants and animals to get the best quality in them. But nowadays we are changing to an area where the genetical editing helps us to get the better quality and best qualities in each and every life forms. Where the life is, we can edit it. We can form a better quality in them. So in 1960s, scientists started bombarding the plants with radiation to cause random mutation in genetic codes. And after that, in 70s and 1974, we have got many developments in these forms, and at last we have got mice that we use in the labs. So these mice are the forms of genetical engineering, where before it the humans were tested. Now the mice are given the test doses for each the medicines, even the foods that are produced, and every chemicals are tested on them. And mostly we need to think about where the discovery of this CRISPR started. Bacteria and viruses, we all know that the viruses and bacteria are the contaminating elements that affects the body. But there is a fight between them. The bacteria and virus been fighting for years until the bacteria become an antivirus. So these phages or bacteriophages, named for the viruses who attacks the bacteria, they start fighting the bacteria and killing them by duplicating the DNAs of the bacteria and making it a factory. So the bacteria became an antivirus by taking and storing the DNA of the RNA of the virus that attacked them, and they stored it. And until further attack came, they collected all the virus DNAs and they searched for the 100% similarity in this RNA, and they are making a new antiviral system which cuts the DNA of the the RNA of the viruses against the attack. And this helped in the discovery of the CRISPR-Cas9. So the protein used there is Cas9. So the Cas9 comes from the fighting of the viruses and bacteria. So the medical applications. We know that we are in the world, the HIV cases is getting more, but we are hopefully thanking God that in India, the mostly less persons who have been positively in HIV is Kerala. And cancer, herbs, these can all, and if as the time is low, I cannot describe it all. So if there is question, I will describe it after. So in HIV, cancer, herbs, the diseases can be eradicated by this CRISPR-Cas9 technology of gene editing. And we all know that most of the people are diabetes, and the stem cells of the skin are taken from the body and they are genetically edited for producing insulin, and they are injected to the persons who is having the diabetes, so they can eradicate the diabetes and they can make the pancreas powerful for producing enough insulin against the glucogenesis of the diabetic conditions. And in ALS and muscular dystrophy or amyottic lateral sclerosis. Sclerosis means it's a nerval diseases where the membrane of the nervous system is getting weakened, and weakening of the nervous system causing the muscular dystrophies and the weakening of the muscles. So here also the CRISPR-Cas9 helped humanity, helped the world for eradicating it. So it's a picture, just look at it on it. So this is how the CRISPR works on the body. First the Cas9 protein forms a complex of the RNA, and it goes and correlate with the matches present in the DNA, and when the RNA gets the information to cut the DNA, it cuts it and take the particular part away from the DNA, and the DNA is mutated. Designer babies. I told that I am focusing on it. So designer babies, we know that the clinical applications, medical application given to a particular person will eradicate his disease and he will die with it. A cured person die with it, but it is not transferred to the coming generation. So designer babies is thinking of a generation where there is no diseases. Whole the generation is eradicated. So it's not a sort of evolution, but it's all the application of the science. So I will talk the process of the producing of the designer babies. So first the oocyte is extracted. Oocyte is extracted from the mother or from the ovary of a female by over hyperstimulation. So in some females there will be no ovarian cycles or something. So by stimulation the ovaries are produced, and the oocytes collected are fertilized outside the body. So maybe the projection of the sperm into it, or by intracytoplasmic